Last chapter, Excretion and Homeostasis. Excretion is the process by which metabolic waste products are separated and eliminated from the bodies of living organisms. This includes carbon dioxide, nitrogenous waste, and excess water. Excretion is a vital process in all living organisms because it prevents the buildup of waste materials in cells, some of which could be toxic, and also creates room for uptake of other materials needed by the cells. In most simple unicellular organisms like the amoeba, excretion occurs through simple diffusion across their plasma membranes. These organisms usually live in aquatic environments. When the concentration of waste materials increases in their cells, they diffuse out into the surroundings. Excess water is usually eliminated through a structure called the contractile vacuole. Amoeba and paramecium live in aquatic environments where the surrounding water is hypotonic relative to their cytoplasm. They therefore experience excessive inflow of water into their cells. This excess water and dissolved chemicals accumulate in the contractile vacuoles. Once full, the vacuole moves to the cell membrane where it bursts, releasing its contents, and other vacuoles are formed. Excretion in plants. Now, in plants, there are no complex organs for excretion of waste materials, and the reason is that most of the materials can be reutilized by the same plant. For example, carbon dioxide produced during respiration can be used for photosynthesis, and the oxygen produced during photosynthesis can be used for respiration. Also, plants are not as active as animals, so the rate of metabolism is not that high. Carbon dioxide, oxygen, and excess water from plants is lost through the stomata and lenticels. Some plants store waste in their tissues like leaves, usually in non-toxic forms. These tissues age and drop off from the plant, taking away the wastes. Excess salts present in plants get deposited in the form of crystals in the cells. Several plant products like gums, resins, tannins, alkaloids like morphine and quinine are also plant waste. Caffeine is stored in coffee berries and tea leaves. It is taken as a mild stimulant to increase mental activity and reduce fatigue. Quinine is a waste product stored in the bark of cinchona tree and aloe leaves. Humans use it for treatment of malaria. Cocaine is produced from leaves of cocoa plant. It is used as a local anesthetic. If taken in large quantities, it causes great physical and mental defects and addiction. Cannabis is stored in fruits, flowers, and leaves of cannabis sativa or bang. It is extracted and used in the manufacture of drugs. Nicotine is found in leaves of tobacco plants and is used in the manufacture of insecticides and narcotic drugs. Rubber is made from latex rubber plant. Colchicine is an alkaloid derived from a plant that is used in genetics, in plants and animals breeding research, and in cancer therapy. Excretion in animals. Now, animals are more active and hence have a higher rate of metabolism. Consequently, waste materials accumulate faster than in plants. Animals therefore have elaborate and specialized organs to carry out excretion. These organs include nephridia in annelids, Malpighian tubules in insects, gills, lungs, liver, skin and kidney in vertebrates. Excretion in mammals. The excretory organs in mammals like humans include the skin, lungs, kidney, and the liver. The skin. The skin is the largest organ in the body, extending from the exterior to the walls of the internal organs like the alimentary canal. It is also called the integument. It is thickest in areas exposed to frequent friction like palms of hands and soles of feet, and it's thinnest in the delicate areas like the eyelids. The role of the skin includes protection against entry of microorganisms into the body, physical damage and the UV radiations, regulation of body temperature, synthesis of vitamin D, excretion of salts, excess water and traces of urea, sensation to pain, touch, heat and pressure, and also fat storage. Structure of the skin. The skin consists of two main layers, the epidermis and the dermis. The epidermis is the outermost layer consisting of mostly keratinocytes, which produce keratin, a protein that gives strength to the skin. The epidermis is divided into 5 zones or strata: stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, and stratum basale. The corneum and lucidum zones are collectively called the conified layer. The granulosum and spinosum make up the granular layer, and the stratum basale make up the Malpighian layer. The Malpighian layer or stratum basale is the deepest epidermal layer separating the epidermis and the dermis. It consists of a single layer of cuboidal cells. This is a zone of cell division and gives rise to the other layers of the epidermis. It is occupied by 3 types of cells. Keratinocytes are the most abundant cells in the epidermis and are found in all layers. The stem cells are found in this layer. They actively divide and regenerate new cells and hence replace the cells shed off at the skin surface. Melanocytes are scattered amongst the keratinocytes. They produce and store melanin that screens off the UV radiations. Merkel cells are few in numbers and give the skin sensitivity to touch. The granular layer, specifically the stratum spinosum, consists of the keratinocytes that are non-dividing and Langerhans cells that help initiate an immune response against pathogens and cancer cells. At the stratum granulosum, the keratinocytes secrete and fill themselves with keratin. This results in disintegration of their nucleus and organelles. These cells are dead but are still functionally sound. The cells divide in the stratum basale and migrate upwards, becoming progressively flatter and keratinized until they get to the conified layer. They are continuously shed off and replaced by new arriving cells. This renewal cycle takes about 30 days. The dermis is thicker than the epidermis. It consists of blood and lymphatic vessels, nerve endings, sweat glands, hair follicle, and the sebaceous glands. Blood vessels ensure nourishment to the skin cells and removal of metabolic wastes. Lymphatic vessels drain the excess tissue fluid. Nerve endings provide sensitivity to heat, touch, pressure, and pain. Sweat glands are made up of coiled tubule of secretory cells which extend into long tubules that open on the surface of the skin as sweat pores. The secretory cells in the cold tubule absorb excess water, mineral salts, traces of urea, lactic acid and carbon dioxide from the surrounding blood vessels and tissues. These substances are secreted in the tubule lumen to form sweat, which flows through the sweat duct to the skin surface. Sweat glands are involved in the body temperature regulation through loss of excess heat by evaporation of water. The hair originates from a deep infolding of the epidermis that forms hair follicle. Sebaceous glands are attached to the follicle. The gland opens into the follicle. They secrete sebum, which keeps the hair and epidermis flexible and waterproof. Sebum contains antiseptic substances for protection against bacteria. The subcutaneous layer is the layer of fat beneath the dermis and binds the skin to muscles and other organs deep in the body. It acts as a storage region for fats and insulation against heat loss. Next excretory organ is the lungs. In mammals, birds, reptiles and amphibians, carbon dioxide formed during tissue respiration is removed from the body by the lungs. In all these groups of animals, the lungs are made up of small air sacs called the alveoli. The alveoli have a thin elastic single layer of epithelium. Below this layer is a dense network of blood capillaries. Blood capillaries around the alveoli have a higher concentration of carbon dioxide than that in the alveoli space. This creates a concentration gradient that causes carbon dioxide to diffuse out of the blood into the alveoli. The carbon dioxide is then expelled from the lungs through a process of exhalation. Next organ, the kidney. The kidneys are a pair of dark red, bean-shaped organs that are part of the urinary system. Each is about 10 centimeters long, 5 centimeters wide, 2.3 centimeters thick, and with an average weight of 120 to 170 grams. The major roles of the kidney are excretion, osmoregulation, ionic balance, and pH regulation. Each kidney is supplied with blood by the renal arteries, while the renal veins drain the blood from the kidneys. The kidneys are located in the abdominal cavity just below the diaphragm. Their relative position is asymmetrical. The left kidney is placed a little higher than the right. The outer surface of the kidney is convex and the inner surface is concave. The inner side has a depression called the hilus, through which the blood vessels and ureter join the kidney. Internally, the kidney structure reveals 3 zones: the outer cortex, the middle medulla, and the inner pelvis. The other components of the urinary system include the ureter, urinary bladder, and the urethra. Urine is formed in the kidney through filtration of blood. Within the cortex and extending into the medulla are millions of tiny filtration units called nephrons. The nephron is the functional unit of the kidney. Each nephron consists of a glomerulus and a system of tubules. The glomerulus is a unique high-pressure system of capillaries that filter the blood. It is encased in a thin double-walled capsule called the Bowman's capsule. The space between the capsule and the capillaries is called the Bowman space. The nephron tubule is divided into 4 segments, and the filtrate passes through each one of these segments before reaching the ureter. A highly coiled segment called the proximal convoluted tubule, which drains the Bowman's capsule, and where almost complete reabsorption of nutritionally important materials takes place. A thin looped structure called the loop of Henle, which reabsorbs water and ions from the urine and plays a role in controlling the concentration of urine. A distal coiled portion called the distal convoluted tubule, which regulates potassium, sodium and pH, and where further dilution of urine takes place. And the collecting tubules, which join with several tubules to collect the filtrate and where final sodium-ion regulation takes place. Each nephron also has a fine network of capillaries called the peritubular capillaries, which is formed by the efferent arteriole leaving the glomerulus. A minute vessel from this network of capillaries forms a U-shaped structure called the vasa recta, which lies parallel to the loop of Henle. These capillaries unite to form the renal vein, which joins the inferior vena cava. Urine formation involves ultrafiltration, tubular reabsorption of electrolytes and nutrients, and secretion of waste materials. Blood flows into the glomerulus through the efferent arteriole and leaves through the efferent arteriole. The afferent arteriole is wider than the efferent arteriole to create high pressure within the glomerulus. Plasma-like fluid is filtered from the capillary blood into the Bowman space. This is called the glomerulus filtrate and forms the primary urine. The process is called ultrafiltration. The glomerular filtrate consists of substances of low molecular size, including water, urea, glucose, salts, and amino acids. Large molecules like proteins and blood cells are not filtered because they cannot pass through the capillary walls. The filtrate in the Bowman space moves into the tubular segments of the nephron. The filtered blood moves through the efferent arteriole into a network of capillaries that surrounds the tubular segment of the nephron. As the filtrate moves through the tubular segment, most of the filtered substances in the glomerular filtrate useful to the body are selectively reabsorbed back into the blood. Equally, some materials may also move from the blood into the renal tubules. In the proximal convoluted tubule, all the glucose, amino acids, some water, and mineral salts are actively reabsorbed against a concentration gradient, a process that requires energy. For efficient reabsorption, the proximal convoluted tubule is adapted in the following ways. The cells lining the tubules have numerous mitochondria which provide the necessary energy in form of ATP. Cells in the tubule have microvilli which increase the surface area for absorption. The tubule is long to provide a large surface area for reabsorption. The coiling of the tubule reduces the speed of flow of the filtrate, thereby giving more time for efficient reabsorption. The tubule is well supplied with blood capillaries. The loop of Henle forms an area where salts, especially sodium chloride, are reabsorbed. The U-shaped loop is generally longer and has a countercurrent flow established between the flow of filtrate and the large supply of blood in the vessels. Active transport is involved in the reabsorption of sodium salts. To regulate the intake of sodium salt, a hormone called aldosterone is secreted by the adrenal glands. Low content of salt in blood stimulates the adrenal glands to secrete more aldosterone hormone, and therefore more salt is reabsorbed from the filtrate and vice versa. The loop of Henle is followed by the distal convoluted tubule, where conditional reabsorption of water and sodium occurs. This region helps to maintain the sodium-potassium balance and blood pH by reabsorbing bicarbonates and selectively secreting potassium and hydrogen ions as well as ammonia. The distal convoluted tubules of several nephrons open into the collecting duct, which extends from the cortex of the kidney to the inner parts of the medulla. The duct helps to reabsorb water, thus increasing the concentration of urine according to the body's state of hydration. It also maintains osmolarity by allowing small amounts of urea to pass through. Further, it also selectively secretes hydrogen and potassium ions and maintains the ionic and pH balance of the blood. And so, the nephron filters blood, reabsorbs substances that are needed, and excretes the rest as urine. Henle's loop and the vasa recta are the part of the kidney that helps to concentrate the urine. A countercurrent flow is created in this area. This is because the direction of flow of filtrate in the loop of Henle and the blood flow in the vasa recta are opposite. This countercurrent flow maintains an increasing osmolarity in the inner medullary interstitium. This countercurrent flow helps to maintain a high concentration gradient and thus effective reabsorption. The kidneys open into the ureter through the renal pelvis. Each ureter is about 25 centimeters long and carries urine from the pelvis to the urinary bladder. The urinary bladder is a hollow, muscular and elastic organ that temporarily stores urine until it's released from the body. Urine leaves the urinary bladder by the urethra, a tube through which urine is discharged. The urethra is enveloped by the urethral sphincter, which controls the flow of urine from the bladder to the outside. Next organ, the liver. Now, the liver is the second largest organ after the skin, lying immediately beneath the diaphragm. It is both structurally and functionally complex. The liver is the only organ in the body that has a dual blood supply, one venous and one arterial. The hepatic portal vein, which carries blood from the spleen, pancreas, and the intestines, contributes about 75% of the blood supply to the liver. This blood is rich in nutrients and other absorbed molecules, but poor in oxygen content. The hepatic artery, a branch from the celiac trunk from the abdominal aorta, contributes about 25% of the liver blood supply. This blood is oxygen-rich but nutrient-poor. Terminal branches of the portal vein and artery join to form the common capillary beds. Thus, a mixture of venous and arterial blood bathe the liver cells called hepatocytes. The functional units of the liver are the cylindrical structures called lobules. Each lobule is organized around a central vein that empties into the hepatic vein and from there into the inferior vena cava. As blood travels through the liver, cells called Kupffer cells remove waste products such as bacteria, old and defective blood cells, and other foreign materials. The breakdown of old and defective red blood cells results in formation of a pigment called bilirubin. The liver has many functions, including bile formation. Bile is formed in the hepatocytes. It is essential in the digestion of lipids because it causes emulsification of fats. Glucose metabolism. The liver converts excess glucose into glycogen, which is stored in the hepatocytes. When glucose is needed, the liver converts the stored glycogen into glucose and is released into the blood. Excess amino acids and proteins cannot be stored in the body. They are broken down by removal of the amino group, a process called deamination. In this process, glucose and ammonia are formed. Ammonia is toxic and is quickly converted to urea, which is less toxic. Other functions of the liver include detoxification of chemicals, thermoregulation, hemoglobin elimination, and regulation of plasma proteins. Diseases of the liver. Now, interbiliary hepatic disease disrupts the flow of bile through the liver, causing a stoppage or slowdown of the bile called cholestasis or biliary cirrhosis. Bile is unable to drain from the hepatocytes. The liver becomes enlarged and takes on a green hue due to bile buildup. Cirrhosis is the end stage of a chronic liver disease when much of the functional liver tissue has been replaced by a fibrous tissue. Jaundice or ictera is the yellowing of the membranes of skin due to buildup of bilirubin in blood. It occurs when the liver is unable to clear normal amounts of bilirubin from blood. Now we move to homeostasis. Now, an organism lives in an environment that keeps on changing in many of its factors, for example, temperature. Similarly, changes occur within the body of an organism due to different metabolic activities. Many of these changes disrupt the biological reactions being carried out within the organisms and lead to adverse effects on the health of the organism. Homeostasis is a process that adjusts these changes in the body of an organism to optimum standards or levels and therefore brings about a steady state. Principles of homeostasis. Various body systems such as the circulatory, excretory, endocrine, and nervous work in a coordinated way to bring about homeostasis. In order to maintain a steady balance in the internal environment, these systems work on a feedback mechanism. There are two types of feedback mechanisms: negative and positive feedback mechanisms. Negative feedback mechanism. When a factor in the body, such as temperature or blood sugar levels, drop below or shoot above the normal, it is detected and corrective action is taken. Such an action is either an increase in level if it is dropping, or a decrease in level if it's decreasing. In positive feedback, a change below or above the normal is not corrected but enhanced instead. For example, during the menstrual cycle before ovulation, small amounts of estrogen are secreted from the ovaries. This estrogen stimulates the release of gonadotropin-releasing hormone from the hypothalamus and luteinizing hormone from the anterior pituitary. The luteinizing hormone in turn stimulates the release of additional estrogen from the ovaries. the levels of gonadotropin-releasing hormone and the luteinizing hormone increase in blood due to this positive feedback effect. After ovulation, the corpus luteum is formed and begins to secrete progesterone in response to luteinizing hormone. Progesterone inhibits the release of gonadotropin-releasing hormone from the hypothalamus and the luteinizing hormone from the anterior pituitary. Blood levels of these hormones decrease because of this negative feedback effect. Thermoregulation. This is the maintenance of a constant body temperature in hot and cold weather. The body has a remarkable capacity for regulating its internal temperature at around 37 degrees Celsius when the temperature rises and falls within tolerable margins. The hypothalamus is the processing center in the brain that controls body temperature. It does so by signaling effectors such as sweat glands and muscles in various parts of the body. When the body temperature goes out of the required range, the hypothalamus sends signals to the skin, which has two ways, either to increase or decrease heat loss from the body surface. When cold, the erector pili muscles contract, making the hair follicles erect. This way, the hairs trap air over the skin, which insulates the body against heat loss. If the body is too hot, the muscles relax, the hair follicles lie down, they trap no air and therefore increases heat loss. Secondly, if the body is too hot, sweat glands secrete sweat which evaporates, carrying with it heat from the body. This results in cooling. Sweat secretion stops when the body temperature falls to normal. Thirdly, when the body temperature is too high, blood vessels near the skin vasodilate and a lot of blood flows near the skin surface, resulting in heat loss. If it's too cold, the vessels constrict, blood flows deep into the body and preserves heat. Skeletal muscles can also receive signals when it's too cold. This results in shivering. The rapid contraction of muscles during shivering results in heat being produced. Osmoregulation. Now, the amount of water and dissolved substances determine the osmotic pressure of blood. The body gains water through drinking, food, metabolic water, and loses the water through sweat, feces, urine, and exhaled air. Osmoregulation attempts to maintain a constant amount of water and electrolytes in blood. When water levels fall in blood, it is detected by receptor cells in the hypothalamus. These receptors are called osmoreceptors. In response, the cells secrete a signaling molecule to the pituitary gland to secrete antidiuretic hormone or the vasopressin. The ADH is released from the pituitary and travels to the kidney where urine is made. Nephrons in the kidney filter urea, salts and other solutes in blood. The last part of the nephron, that is the collecting duct, is the final place where the body can absorb water. The ADH binds to receptors, triggering a series of reactions that lead to increased permeability of the tubular walls to water. This allows more water to move back into the bloodstream. This will ultimately result in a highly concentrated urine. Regulation of ionic content. Other than regulating the amount of water in blood, blood pressure is also regulated by the ionic content in blood. Ions, particularly sodium and chloride, play a vital role in blood pressure regulation. Ion regulation is done by the renin-angiotensin-aldosterone system. A drop in blood pressure will be detected by the kidney, and in response, the kidney releases the enzyme renin. This enzyme activates the angiotensinogen hormone to angiotensin 1 in blood. Angiotensin 1 travels to the lungs where it is converted to angiotensin 2, the active form. It has short and long-term effects. First, it triggers vasoconstriction, hence raising the blood pressure even at lower blood volume. Angiotensin 2 also signals the adrenal glands located above each kidney to secrete the hormone aldosterone. Aldosterone signals the kidney tubules to retain sodium and chloride ions and water. In fact, it is the retention of sodium and chloride ions that raise the osmotic pressure of blood, causing water to be reabsorbed through osmosis. Blood volume thus goes up. When pressure is restored, the signaling cascade stops and normal urine is formed. So the renin-angiotensin-aldosterone system, together with the ADH, help in osmoregulation through control of blood water and ionic content. When the pituitary cannot produce the ADH totally or produces it in inadequate amounts, reabsorption of water from the kidney tubules does not happen. A lot of water is lost in urine. This results in large volumes of dilute urine. The individual feels thirsty and dehydrated more often and compensates that by regular intake of water. This is called diabetes insipidus. And finally, blood sugar regulation and diabetes mellitus. Blood sugar or glucose is the fuel that the body uses for energy. But this glucose cannot be utilized without insulin, a hormone produced by the pancreas. Diabetes mellitus is a condition in which the pancreas does not produce insulin or the body does not respond to insulin produced. Food is made up of carbohydrates, proteins, and lipids. The digestive system breaks up carbohydrates into glucose, which is absorbed into the bloodstream. The pancreas results to the elevated blood sugar by releasing insulin. Insulin is like a key that opens body cells, allowing glucose to enter. As blood sugar enters the cells, the levels fall in blood. If you have type 1 diabetes, the pancreas does not produce insulin, so glucose builds up in blood. Remember, without insulin, blood sugar has no way of getting into the cells. In type 2 diabetes, the pancreas does not make enough insulin. In addition, the body does not respond to insulin properly, so much of the sugar remains in blood. If left unchecked, both type 1 and type 2 diabetes mellitus lead to dangerous elevation of blood sugar. This causes high blood pressure. Over time, persistent high blood pressure can lead to kidney failure, heart failure, blindness, or even amputations. The symptoms of diabetes include passing large amount of urine, feeling of thirst, presence of high levels of glucose in urine, loss of body mass, dizziness, and blurred vision. Diabetes is controlled by regular administration of insulin hormone to help lower the blood sugar, avoiding intake of foods rich in sugar, alcohol, and salts.